Welcome everyone. Thank you for joining our webinar. Dr. Selina will be giving our presentation today. She is the field application specialist of IVIM technology. Her presentation will be on the theme In Vivo Application, in Vivo Microimaging for Immunology. We are very sorry that this webinar will be given by recording due to the time difference. During the webinar, if you have any question, please text it in the Q&A box or send email to us. We will respond to your questions via email as soon as possible. I will hand it over to Dr. Selina.
Hi, this is Selina. I'm an application specialist in IVIM technology. Today, I will briefly introduce our technology and applicational examples of various organ imagings for immune research in vivo using IVIM products. Firstly, I'd like to introduce IVIM's intravital microscopy to observe live tissue and cells in live animals. While X-ray, CT, and MRI imaging technology provides only static and tissue-level information in a living object, intravital microscopy can provide a detailed, live cell-level video of disease development as illustrated in this example video. Intravital microscopy is an extremely powerful tool that enables imaging several biological processes in live animals in long-term manner. Conventionally, for intravital experiments, users have to prepare each required functions by themselves. There was a high degree of technical difficulty and non-optimized performance with limited applications to imaging interest organs. It was very difficult and it's not an optimal solution with low performance without any standardized in vivo protocols. Our all-in-one intravital confocal and two-photon microscopy system is fully optimized for intravital live cell imaging. IVIM is the world's first and only one company providing a new all-in-one system with the best intravital imaging performance. We provide various accessories for wide range intravital applications with strong technical support for various field applications. All-in-one single box package enables easy installation, operation, and maintenance. It has co-optimized hardware and software for superb intravital imaging performance with ultra-fast imaging speed and no limitation in imaging various internal organs. Together with live tissue motion compensation function, you can get the best quality in vivo data. With ultra-fast video rate up to 100 frames per second, thereby we can even capture the fast dynamics such as red blood cells that are flowing inside of the blood vessels. We can get 4-color, confocal, and two-photon imaging with submicron resolution, which finally enables real-time visualization of live cell dynamics such as microcirculation and cellular physiology in vivo. IVIM's intravital microscopy is to observe live tissue and cells in live animal, which enables 3D cellular level microscopic imaging and analysis in vivo. Our all-in-one intravital confocal and two-photon microscopy system is fully optimized for intravital live cell imaging, such as real-time cell trafficking, cell-to-cell interaction, and cell-to-microenvironment interaction inside of the living body. Furthermore, it can be widely applied to the therapeutic research fields, investigating drug delivery to the target tissue, drug efficacy, and therapeutic mode of action in various preclinical mouse models of human disease. Here's one example of in vivo familial lymph node imaging. We adaptively transferred T cell and B cells, which were obtained from actin-DS-RAD and actin-GFP transgenic mice. And we monitored the cellular extravasation dynamics of the T and B cells in high endothelial venue of the lymph node in real time. Here, T and B cells are selectively extravasated through the specific sites of the HEV. For intravital lung imaging, as you already know, when we make an incision on the rib, the air pressure will be changed and thereby killing the mouse. So we need to make mouse breathe again by intubation, which will be connected to the ventilator. Together with lung imaging window chamber for stable visualization, we can acquire a clear video of neutrophil dynamics in pulmonary vasculature after sepsis induction by injecting LPS. Then pulmonary neutrophils migrated to the specific sites and block the microcirculation of the lung blood vessels. This is in vivo time-lapse movie of neutrophil infiltration in pulmonary microcirculation. Also, we can measure the blood flow in a real-time movie of RBC flow, and we analyze the differences of the vascular area-specific velocity in pulmonary microcirculation of LPS-treated mouse model. For the application, we have a lot of examples according to the interest organs. Here are some examples of in vivo imaging in various organs such as lung, bone marrow, and skin visualizing inflammatory responses. So I will explain the details in the following slides. In terms of tumor, IVIM system can be applied to the cancer xenograft model, investigating T-cell interaction with tumor cells in vivo, and a cancer metastasis model visualizing circulating tumor cells flowing through the blood vessels. We can perform imaging analysis of the in vivo drug delivery effects to the tumor tissue. IVIM's intravital microscopy has succeeded in high-quality live cell imaging in almost all of internal organs in a live mouse model, as you can see in this slide. It includes brain, brain tumor, skin, lung, pancreas, spleen, GI tracts, prostate, lymph node, memory tissue, placenta, kidney, liver, muscle, heart, and even retina imaging in vivo. IVIM technology provides all-in-one intravital confocal and two-photon microscopic system, which is IVIM-C, IVIM-M, CM, MS2, CMS, and customized system. C is the confocal-only model, M is the multiphoton only but with wavelength tunable laser, and CM is the most combined model of the confocal and two-photon microscopy. IVIM-MS is the most compact and simple version of two-photon with 920 nanometer wavelength fixed laser module. Customized system is fully optimized for various research purposes, including large animal imaging, retina imaging, endomicroscopy, rotating objective system for brain imaging, and so on. In addition, IVIM also provides one-stop total research service of intravital imaging for the researchers in academia and the therapeutic industry. This is a simple introduction video of IVIM CM device, which has both confocal and two-photon system in one device. There's a scanner for X and Y scanning, which has a polygon mirror set and also a galvanometer set for scanning system. And we have some mirror and lens sets and a motorized stage with a temperature control system for live animal models. Then all these systems are together in an all-in-one device. Confocal system enables high-resolution optical sectioning with ultra-fast frame rate by using polygon scanner. Thereby, we can acquire the high quality of real-time movie and multicolor intravital imaging of the live tissue. Finally, with these technical innovations, IVIM's all-in-one intravital microscopy system is capable of high-quality subcellular resolution 3D imaging of in vivo tissue. This particular example is obtained from inguinal adipose tissue of live mouse model by using confocal model, IVIM-C, which clearly shows green adipocyte and red blood vessels in 3D. Two-photon microscopy enables point excitation, then we can get the high-resolution optical sectioning imaging with deep imaging depth, with long wavelength near-infrared pulse laser source. We can acquire the clear images with minimized photofleeting and reduced phototoxicity to the live tissue. In addition, by second harmonic generation, we can detect skeletal muscle structures without additional labeling. This is wide area image we obtained from skeletal muscle of the live mouse model by using two-photon model IVIM-CM, which clearly shows blood vessels in blue and nerves in green. Now let me zoom into this area. In this single field of view image, now you can clearly see the red sarcomeres in myocyte with second harmonic generation signals without additional labeling, and green neuromuscular junction and blue capillaries in tomographic manner. It is more easier to see the whole cellular level distribution in this maximum intensity projection image. I'd like to briefly introduce key technical features of IVIM's product. First of all of IVIM's all-in-one intravital microscopy system is equipped with ultra-fast laser scanner, polygon scanning system, which enables ultra-high-speed Invivo imaging at maximum speed of 100 frames per second with 512 by 512 pixels. Also, it could achieve uniform excitation illumination over the entire imaging field of view. Thereby, we can acquire the high-quality image and videos at ultra-fast frame rate in real time. This is very important to obtain high-quality in vivo image from live animals by compensating the tissue motion artifact. Otherwise, other competitors usually have galvano scanning system, which only can get uniformed excitation due to the unstable scanning speed of resonance scanner over the entire imaging field. Also with limited scanning speed. This is polygonal mirror set assembled in every product lines of IVIM. In addition to ultra-fast imaging, IVIM's intravital microscopy system is fully integrated with animal motion compensation function, which is compensating the motion artifact of live animal model. It enables an automatic hassle-free, high-precision motion compensation imaging by facilitating frame-by-frame motion compensation. By GPU-assisted algorithm processing, we guarantee you to get the clear imaging result of various tissues. It has a synergetic effects together with ultra-fast scanning frame rate. In this side-by-side comparison, this is a movie of kidney. We can see the clear changes before and after applying the function. It is very clear that the high-quality in vivo imaging needs this kind of motion compensation function. Without motion compensation, only this blurred image can be obtained due to the motion artifact, which disturbs the quantitative cellular analysis of imaging data. This is another example of heart imaging. Even though raw imaging data has high degree of movement, by applying motion compensation function, we can acquire the high quality of clear image without any shifting artifact. Please note that a conventional standalone microscopy only can acquire those kind of shifted images like in the middle from the same live mouse model. This is the video actually working in the studio software. Just one click for selecting reference channel for the compensation and activate the motion compensation button. and click capture, then we can directly get compensated result in the display window right away. We are also providing various additional accessories for maintaining a healthy condition of live mouse model during in vivo experiment. Here, we have rectal-proof sensor for mouse body core temperature, which are feedback system with the heating plate, maintaining a stable body temperature throughout the experiments. Also, we provide glass heater for local tissue temperature maintenance of imaging organ or tissue. So if you need to move an organ slightly out of the body to image, then this tissue temperature heater with integrated cover slip that comes in contact to the tissue and it maintains the local tissue temperature. This is the translational stage for mounting the mouse model. Every accessories are connected to the connector in the left, and we can slide in and out of the stage before and after mounting. Also, all of IVIM's Intravital Microscopy has its own operating and post-processing software. The name is IVIM Engine and IVIM Studio. With IVIM Studio, we can visualize the structure of cancer metastasis lymph node upon the imaging depth, including GFP expressing cancer cell, red blood vessel, and blue lymphatic structures. Or we can perform cross-sectional analysis of the cell nucleus and vasculature structures in the villi of the small intestine. And we determine the co-localization of each cells in 3D. The following is the 4D data we acquired by 3D imaging of ear skin over time. 4D analysis of the cell dynamics and cross-sectional colocalization analysis are available in real time using Studio software. With measurement function, quantitative analysis of vessel dilation before and after drug treatment, immune cell trafficking, cell-to-cell distance, and cell-to-cell contacts can be performed in Studio software. In addition, 4D quantitative analysis of immune cell movement, cellular speed, direction, cell-to-cell distance, and the number of contacts over time can be performed in 4D in Studio software. For the application, we have many examples according to the interest organs. This is intravital imaging of ear skin, visualizing immune cell dynamics with LysM GFP transgenic mouse, together with ear skin blood vessels which were labeled by IV injection of PCAM antibody conjugated with fluor-4. As we can see here, we could visualize the cell-to-cell dynamics of neutrophils in skin, and even we can see the very fast dynamics of the flowing neutrophil cells through the blood flow. This is another example of in vivo imaging of Cars protein, the enzymes that control the protein synthesis. To investigate the details of how this enzyme induces inflammatory responses in vivo, we monitor the immune cell dynamics after intradermal injection of Cars protein to ear skin tissue. Time-lapse intravital microscopy showed the active recruitment of macrophage and monocytes to the injected KRS, which enables in vivo analysis of chemotaxis effects of the therapeutic material. This is the result of in vivo imaging of popliteal lymph node, visualizing T cells and vascular endothelial cells in HEV. A is the mosaic image of a whole popliteal lymph node showing the distribution of the T cells in red and vasculatures in green in the cortical side of the popliteal lymph node at 1 hour after the transfer of the T lymphocytes. In magnified images, we can monitor the serial extravasation dynamics of the T cells in HEV. T cells firmly adhere to the endothelial cells, started to transmigrate across the endothelial cells from the lumen of the HEV. This is the result of in vivo high spatial temporal resolution tracking of flowing RBCs and T cells in the post-capillary HEV. Velocity color maps of the RBC and T cells show the changing velocity of the individual flowing cells in HEV. The velocities of both T cells and RBCs dramatically decreased when they encounter to the HEV. Especially three distinctive dynamics of behaviors of T cells, flowing, rolling, and sticking were clearly observed. Moreover, to clearly visualize the stepwise dynamics of the post-luminal T cell migration in HEVs, we adaptively transferred GFP-expressing T cells and injected blue fluorescence labeled anti-ERTR7 antibody to an active DS-RAD mouse expressing DS-RAD in endothelial cell. Results clearly shows the multiple steps of T-cell migration across the HEV, which were composed of endothelial cells and FRCs through the perivascular channel. Firstly, T-cell adhesion to the endothelial cell and intraluminal crawling, trans-EC migration, intra-PVC crawling, and trans-FRC migration, then finally arriving to the lymph node parenchyma. In addition, longitudinal 3D monitoring of the same sites in HEV reveals that T cells transmigrated through the preferred site in AC and FRCs of HEV. Next, to observe trans-EC and trans-FRC migration hotspots for endogenous lymph node homing cells, we used Kaede transgenic mice, in which all cells express photoconvertible fluorescence protein Kaede. When a 405 nanometer laser was irradiated onto the HEVs, all cells in an A field of view changed from green to red, and newly appearing cells in the HEV lumens are green. Therefore, we were able to observe the trans-EC and trans-FRC migration of the green cells across the red photoconverted ECs in HEVs. Up to 12 and 8 green cells transmigrated across ECs and FRCs respectively at the same sites in HEV during 1.5 hours. Collectively, mouse of the adaptively transformed model and the Kaede mouse clearly show the existence of the trans-EC and trans-FRC hotspots of T and B cells migration in HEVs. Furthermore, it is observed that many T cells transmigrated across FRCs at the same sites covered by perivascular disease. To determine whether T cells transmigrate across FRCs, which were covered by disease, preferentially or accidentally, we compare the percentage of the trans-FRC migration sites covered by disease with disease coverage on HEV. The percentage of the trans-FRC migration sites and the hotspots covered by disease were significantly higher than the disease coverage on HEV. Collectively, these results reveal that T and B cells preferentially transmigrate across the FRCs which were covered by disease, and perivascular disease may regulate the trans-FRC migration of T and B cells in HEV. Aside from in vivo imaging, optical tissue clearing technique enables 3D deep tissue imaging of intact lymph node, visualizing the three-dimensional cellular distribution of the T and B lymphocytes in the lymph node at the specific time points. In addition, we can perform in vivo retina imaging by utilizing additional optic system. This is the result visualizing various immune cells in mouse retina, including macrophage, retina ganglion cells, microglial cells in vivo using various transgenic mouse models which are expressing fluorescence proteins in the specific immune cells. This is the real-time movie of microglial cell dynamics in mouse retina. We can analyze the cellular movement and activity of the microglial cells in real time and measure the cellular velocity of dendrite movement in a normal and drug-treated mouse retina. For in vivo longitudinal monitoring, we utilize in vivo imaging windows such as dorsal skin fold chamber for the cancer or drug candidates in vivo analysis, and cranial imaging window for brain imaging, and abdominal imaging window for internal organs. By utilizing dorsal skin fold chamber, we can perform repetitive in vivo imaging of tumor development or in vivo analysis of target drug delivery in the tumor tissue. Firstly, we implant the tumor cells and monitor the development of the tumor on the site. Visualizing together with tumor blood vessels, we can analyze the drugs and anti-angiogenic effects on the tumor tissue. We could find that in the drug-treated group, the tumor vessels are less dilated compared to the normal group. After drug treatment, vessel morphology became more normalized compared to the sham control model. This is another example investigating nanoparticle delivery to the breast cancer cells. Over time after particle injection, we could monitor the delivery rate of nanoparticle to the tumor tissue. Within 24 hours, nanoparticles are fully delivered to the breast cancer tissue. For longitudinal in vivo imaging of brain, we utilized a cranial imaging window. We implanted the cranial imaging window chamber using dental cement on cranial bone as in this picture. Firstly, we make a scalp skin incision, remove periosteum with sterile ear tips, and scrub the surface on the skull and make a circular groove in the skull with drill and remove the excised foam flap. Then cover the dura with the cover glass. Apply UV curable adhesive to nearly dry skull. Then we seal the optical window with dental cement, covering all the exposed skull. Then we place the mouse model on the heating stage, adjust the focal distance. This is one example of in vivo brain imaging utilizing a cranial imaging window. Brain neuroinflammatory responses was induced by 3-NP chemical injection of lysozyme GFP transgenic mouse model. After induction, lysozyme expressing leukocytes are densely located to the specific sites over time. Magnified longitudinal images of 3-NP group showing a dynamic redistribution of the lysozyme-positive cells to the specific sites in cerebral cortex. Also, from the time-lapse imaging, we could visualize and analyze the meandering and crawling behaviors of the lysozyme-positive neutrophils in brain of neuroinflammation mouse model. In addition, to observe longitudinal changes in lysozyme-positive cell infiltration and vascular integrity of BBB in the cerebral cortex, we performed a 4D analysis by repetitive G-stack imaging in the brain over time. To analyze the vascular leakages indicating a BBB integrity, we did IV injection of Evans blue and visualized the blood vessels and monitored the vascular permeability in a repetitive manner over time in 3D. In control model, there was no identifiable changes in vascular morphology and permeability until day 30. However, in neuroinflammation mouse model, vascular leakages were observed in the parenchyma of the cerebral cortex from day 14 to day 21, suggesting a persistent, compromised BBB integrity. At day 30, numerous Lyme-positive cells had infiltrated to the same site with vascular leakages, suggesting in vivo progression of BBB dysfunction to severe neuroinflammation during 1 month. Finally, longitudinal in vivo imaging of cranial imaging window implanted model can be a useful approach to investigate underlying cellular mechanism of BBB dysfunction and neuroinflammation in neurodegenerative disease in vivo. And we utilize abdominal imaging window for longitudinal in vivo monitoring of internal organs such as pancreas, spleen, and kidney. This is the result of in vivo imaging of spleen and pancreas, and we could visualize real-time cellular dynamics of endogenous monocytes and dendritic cells in spleen by using a transgenic mouse. And this is the example of pancreatic islet cells imaging. And islet beta cells in pancreas were visualized in vivo, and their cellular dynamics and the development were monitored in a long-term basis by utilizing abdominal imaging window. After islet transplantation, we monitored the islet cell engraftment and cellular development in kidney in a long-term manner. In addition, in a colorectal cancer model, we tracked the development of the cancer tissue in a cecum and changes in the microenvironment according to the treatment of anti-cancer drugs in the same mouse model together with the vascular changes. Again, in vivo imaging could be the great method to monitor the temporal dynamics of the disease development in the same mouse model over time. IVIM technology has many experimental in vivo research experiences throughout almost all organs in live mouse model without any restriction of the imaging organs. To summarize, IVIM technology is a total solution provider of intravital microscopy technology. IVIM is providing a unique all-in-one intravital microscopy with strong applicational support and high-quality intravital imaging research service for pharmaceutical R&Ds. And we have lots of intravital research publications by using our IVIM system, categorized by imaging organ and imaging purposes, such as red blood cell flow analysis, in vivo validation of drug and cell delivery. This is exemplary project IVIM performed. In this work, IVIM visualized the intravital delivery of exosome to the target cells in sepsis animal model. As you can see in this imaging result, the injected exosome arrived to the target cell only after 10 minutes, then became internalized after 30 minutes. Thereby, if you have any question, according to intravital experiment of your interest organ or the tissue in your mouse model, please feel free to contact us. Then we can fully support you for the best intravital data. Thanks for your attention.